Welcome to today's MedPhysio tutorial on humoral immunity, the arm of adaptive immunity that protects us using antibodies. Think of it as the body's guided missile system, engineered to target threats with remarkable specificity. By the end of this session, you understand not just what antibodies do, but why they are essential for long-term protection, vaccines, and everyday clinical immunity. So imagine a patient who receives a tetanus booster after many years. Within days, antibody levels skyrocket. Why so fast? It's because humoral immunity remembers. This branch of adaptive immunity forms the basis for all major vaccines, drives many diagnostic tests like ELISA, and powers therapeutic monoclonal antibodies. Understanding it is vital for linking physiology to real-world activities. Our learning objectives. First, we'll outline the components and sequential steps of humoral immunity. Next, we'll explore B cell activation and compare T-dependent and T-independent pathways. These are two different instructional modes for B cells, one guided by T cell help and one in which specific antigens can activate B cells directly. Then we move into the architecture of antibodies and the classes they belong to. Hemoglobin M, hemoglobin G, A, E, and B. After that, we'll detail the functions antibodies perform, and finally, we'll connect these mechanisms to clinical examples. As we move forward, please keep this slide in mind. Now, while the previous slide outlined what we'll cover, what we'll be able to demonstrate both conceptually and in an exam setting is listed here. First, be able to define humoral immunity clearly, distilling the entire concept into a precise, elegant statement. Second, you'll be able to describe B cell activation mechanisms. You should be able to sketch it or describe it in a flowing narrative that mirrors physiological sequence. Third, you'll distinguish primary versus secondary immune responses. Understanding this difference is the key to interpreting vaccine behavior and booster schedules. Next, you'll be able to predict which antibody class is involved in various clinical scenarios. Finally, you'll learn how to write well-structured physiology exam answers on this topic. By the end of the session, each of these outcomes should feel achievable and natural, like tools you can reach for with confidence. Let's slow down and build a clear, grounded definition, something that feels both conceptually solid and exam ready. So we start with the central idea. Humoral immunity is the arm of adaptive immunity mediated by antibodies produced by B cells. Think of antibodies as circulating molecular tools, delicate in design, yet highly precise in their ability to identify and neutralize pathogens. Now bring your attention to the phrase, targets extracellular microbes and toxins. This is the key. Humoral immunity works in the spaces between cells, blood, the lymph, mucosal secretion, where bacteria, viruses, and toxins float before they can enter host cells. Next, include the three major cell types involved: B cells, the plasma cells, and the memory B cells. Imagine these as stages in a storyline. Naive B cells wait for their specific antigens. Once activated, they transform into plasma cells, which are the dedicated antibody-producing factories. Now, a subset becomes memory B cells, silent but vigilant, ready to respond far more rapidly on future encounters. Finally, note the phrase requires antigen recognition, with or without the T cell help. This is your cue to appreciate the two possible pathways. One, T-dependent activation, where helper T cells provide critical signals from class switching and affinity maturation. Second, T-independent activation, where certain antigens, often large polysaccharides, can activate B cells directly, though with less precision and limited class switching. Together, these points form a complete functional definition. Let's take it. Humoral immunity is a targeted, antibody-driven defense that operates in the extracellular space, shaped by the coordinated actions of B cells and often T helper cells. Beautiful. Hold this definition gently in your mind. It's the foundation for everything that follows next. So let's about B cell activation using the T-dependent pathway. Imagine a B cell meeting a protein antigen for the first time. B cell receptor binds to the antigen. That first contact is enough to pull the antigen inside. The B cell then breaks it down into small peptides and displays one of these pieces on the major histocompatibility complex, class 2, MHC2. Almost like raising a flag that says, "Can someone confirm this?" A nearby CD4 T helper cell, already primed to that same antigen, notices the MHC2 peptide complex. Once it recognizes the match, the T helper cell becomes activated and provides the help the B cell needs. So it guides the B cell to move beyond its basic immunoglobulin M response. Immunoglobulin M is the antibody produced by the B cell. It's the first line of defense in the immune response, and it acts by agglutinating pathogens and activating the complement system. Now, B cell, with the help of the T helper cell, the B cell undergoes class switching, which means it changes from using immunoglobulin M to other types of immunoglobulins. That's the immunoglobulin G, A, or E, depending on the cytokine signals. At the same time, affinity maturation occurs in germinal centers. This helps to sharpen the antibody specificity, so the B cell produces high affinity antibodies with much greater precision. Now, from an exam perspective, remember that this T-dependent pathway is the high quality, premium version of humoral immunity. It produces not just the right class of antibody, but a much better one. Higher affinity, more functionally appropriate, and capable of sticking around as long-lived plasma cells and memory B cells. That longevity is why protein-based vaccines work so well. The T helper cells drive the formation of memory cells that can last for years. Binding, activation, class switching, affinity maturation, production of memory B cells, and plasma cells last for a very long time. Now let's look at the more straightforward, faster pathway. The T-independent response is what happens when a B cell encounters an antigen lacking protein component like a polysaccharide capsule or specific lipid-rich bacterial structures. These antigens have repeating molecular patterns on their surface. As a result, they can pull together and cross-link many B cell receptors simultaneously. That mass clustering delivers a strong but very basic activation signal, enough to turn the B cell on, but not sophisticated enough to involve the T helper cells. Once activated, the B cell proceeds with what it can do on its own. It proliferates, differentiates, and begins secreting antibodies. But because there is no T cell help, the cell does not undergo class switching or affinity maturation. So the antibodies released are mainly the immunoglobulin M, right? It can't be class switching and change to other immunoglobulins. So the antibodies released are mainly immunoglobulin M. These are quick to appear, broad in action, but not highly refined. As the response settles, something important becomes clear. Without the T cell involvement, the immune system cannot build strong, long lived memory. A small number of memory like cells may survive, but they lack the durability and precision seen in T dependent immunity. Let's picture the antibody as a neat Y shaped molecule. It's simple at first glance, but incredibly versatile. Each antibody is made of two heavy chains forming the sturdy core of the Y and two light chains sitting along the outer edges. Together, they shape the two key regions you must never forget. At the tips of the Y are the Fab regions, parts that recognize and bind an antigen. You can think of them as the fingers of the molecule. They're highly variable and tuned for precision. At the base is the FC region, which is much more constant and it's like a communication hub. This is the part that calls in help by activating complements, engaging phagocytes, or directing the immune response. Now, a good exam phrase here is Fab determines specificity, FC determines the effector response. Now, all antibodies fall into five major classes, and each class is designed for a different type of defense. Immunoglobulin M is the first to arrive, often forms pentamers, making it excellent for early agglutination and rapid containment of infection. IgG is the most, globulin G is the most abundant antibody in the blood and tissues. It provides long-term high affinity protection and is the only class that crosses the placenta. Immunoglobulin A patrols the mucosal surfaces, saliva, tears, the gut, and it serves as the frontline guard against pathogens that try to enter. Immunoglobulin E is rare but powerful, driving responses to parasites and triggering classic mast cell mediated allergic reactions. The immunoglobulin D, found mainly on naive B cells, functions as part of the signaling machinery that helps those cells recognize antigens and begin activation. Let's think of antibodies as quiet, precise guardians, shown built to intercept a threat before it can spread. When we talk about the mechanisms of antibody action, we're really describing how these proteins turn recognition into protection. The first mechanism is neutralization. Here, the antibody covers the harmful parts of a virus or toxin. That's the very region it would typically use to enter or damage the cell. By masking those sites, the antibody makes the pathogen biologically harmless. Next is opsonization. That's making it tasty, which you can picture as placing a bright marker on the enemy. When antibodies bind to a microbe, they expose their FC regions as handles. Remember, FC, the Y shape. Phagocytes such as macrophages and neutrophils recognize and bind these FC portions via their FC receptors, allowing them to engulf the target more efficiently. Then we move to The complement activation is the third mechanism. Here, immunoglobulin G or immunoglobulin M binding triggers the classical complement pathway. Complement proteins assemble in a cascade, amplifying one another until they form membrane complexes that punch holes in the pathogen's membrane, resulting in its lysing. So they lyse the pathogen. Finally, consider the ADCC, that's the antibody-dependent cellular cytotoxicity. In this pathway, antibodies bind larger targets such as virus, infected, or tumor cells. Natural killer cells scan for Fc regions facing outward, latch onto it, and release perforin and granzymes, triggering apoptosis of the target cells. It's a precise, contact-dependent form of immune policing. Now, for your exams, the perfect summary is neutralize, opsonize, complement, ADCC. These four words map directly to the physiology, and you score full marks every time you answer in this fashion. Imagine encountering a pathogen for the very first time. The immune system approaches with caution, almost like noticing a stranger in a crowded room and taking a moment to decide whether they're a threat. This is the primary immune response, and it naturally unfolds more slowly. Naive B cells need time to activate, multiply, and start producing antibodies. So the earliest antibodies are mostly immunoglobulin M. They are large, efficient, first responders that appear within days, not immediately. The affinity is modest because the immune system hasn't yet fine-tuned its recognition. Now compare that to the secondary immune response. This feels more like spotting a familiar face. Here, memory B cells that were generated during the first encounter are already primed. Once they recognize that same antigen again, they respond with remarkable speed and precision. Antibody production rises within hours to a couple of days. At this time, immunoglobulin G dominates. These immunoglobulin G molecules have undergone affinity maturation, so they bind more tightly and more specifically. It's the immune system saying, "Ah, I remember you. I know exactly how to neutralize you." Now, from an exam perspective, the difference comes down to speed, isotype, and quality. In the primary response, it is slow, it is immunoglobulin M dominant, and there's low affinity. While in the secondary response, it is rapid, it is immunoglobulin G dominant, and has higher affinity. This distinction forms the physiological basis for booster vaccinations. By reintroducing an antigen, booster reawakens memory cells, creating a swift, high affinity immunoglobulin G surge that reinforces long-term protection. Now let's discuss how you structure um, the answer for an exam question on this topic. The structure mirrors the natural logic of the immune response. You can begin with a definition. The tight, confident opening signals mastery right away. For example, humoral immunity refers to antibody-mediated adaptive defense in which B cells recognize antigen and differentiate into plasma cells that secrete antigen-specific antibodies. Beautiful. Next, state the location and key cells. Briefly mention where these events take place. This includes the secondary lymphoid organs and germinal centers. Then highlight the central players, the B lymphocytes, T helper cells, and antigen presenting cells. This gives your answer a clear physiological angle. Then move into the mechanism, activation, proliferation, and differentiation. Walk through it in a clean sequential order. Activation. Here, antigen binding to the B cell receptor plus T helper cell involvement where required. Next, proliferation is clonal expansion of antigen-specific B cells. Differentiation. Here, there's emergence of plasma cells that secrete antibodies and memory B cells that persist. Now, connect these to antibody class functions. Describe how immunoglobulin M dominates the early phase. Immunoglobulin G provides long-term systemic protection. Immunoglobulin A guards mucosal surfaces. Immunoglobulin E responds to parasites and allergens. And D serves primarily as a membrane receptor. These transforms facts into a functional narrative. Afterwards, add clinical correlations. The vaccine boosters and immunoglobulin G, immunoglobulin A deficiency, and recurrent mucosal infections, or the immunoglobulin E driven allergic responses. These elements show that you understand not only the mechanisms but also their real world significance. You can also finish with the significance of the memory response. It is faster, creates a more faster, more robust, and has a higher affinity antibody production upon re-exposure. Now, exam tip, always mention the T helper cell involvement in T-dependent responses. Examiners watch for this because it shows you understand how B cells receive instructional signals for class switching and affinity maturation. That's the T helper cell involvement in T-dependent responses. Common mistakes that students make, they usually forget to contrast the primary versus the secondary antibody responses. A quick line about timing, antibody class patterns, and affinity differences can instantly elevate your answer. The key takeaways from this tutorial. Humoral immunity is the arm that produces antibodies. Those antibodies circulate like storytellers, past encounters, turning recognition into protection. A key distinction to hold on to is the difference between the T-dependent and the T-independent responses. When a B cell receives help from a T helper cell, it enters a much richer developmental pathway, which includes class switching, high affinity refinement, and strong memory. Without that T cell help, the response can still occur, but it remains limited, mainly producing immunoglobulin M with weak or short-lived memory. Each immunoglobulin class then fits into its own functional niche. Immunoglobulin M rises early and activates complement. G provides high affinity long-term defense. A protects mucosal surfaces. And E arms mast cells for rapid responses to parasites and allergens. In exams, anchoring each class to its signature role usually earns good and easy marks. When antibodies engage a pathogen, they use a familiar set of maneuvers. They neutralize by blocking binding sites, they opsonize to mark targets for phagocytes, and they activate complement to amplify inflammation and destroy microbes. Those three actions, neutralize, opsonize, complement, capture most of their protective work. And finally, The secondary immune response stands out as the system's proof of memory. After first exposure, memory B cells wait quietly. On re-exposure, they trigger a response that is faster, stronger, and dominated by high-affinity immunoglobulin G. This is the physiological logic behind booster vaccines. They rehouse the system so protection remains sharp. If you keep these things in mind, how antibodies work, how class switching and memory arise, and why secondary responses are so much more powerful, you have the clearest map for any question on humoral immunity. Thank you for joining this MedPhysio session. Keep exploring, stay curious, and remember, physiology becomes easy when you understand the why behind every mechanism.